Welcome to Humanized Mouse Models for Biomedical Research, Selection and Experimental Implications. I'm Tamlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and I'll be the moderator for this webinar. Ethical and technical constraints severely limit the in vivo study of human immunity in mice. As a result, there's a significant need for discourse as well as novel approaches. Today's webinar will focus on the strengths and weaknesses of current humanized mouse models, introduce new strains, and explore the use of humanized mice for infectious disease, cancer, diabetes, and drug testing research. Our first speaker is Michael Brehm, Assistant Professor, Program in Molecular Medicine at the University of Massachusetts Medical School. Dr. Brehm will discuss the creation of humanized mice to study human immune responses and outline considerations for researchers in choosing the most appropriate models for their studies. Dr. Brehm will be followed by Charlie Miller, Director of Customer and Technical Support at the Jackson Laboratory. Charlie will discuss work being done at the Jackson Laboratory. But before they get started, I want to encourage you to submit questions for our Q&A session at the end of the presentations. We'll answer as many questions as we can. Simply type your question into the Ask a Question box and hit Submit. Okay, Michael, we are ready for you to get this webinar underway.
Thank you, Tamlin. It's a real privilege to be here today and to basically describe some of our work on the development of humanized mouse models to study human immunobiology. And what I intend to accomplish today is to basically give you an overview of humanized mouse models, starting off with a basic description of the cutting-edge mouse strains we use to create humanized mice, and then basically describe how we use these model systems to study human immune responses and human immunity in an in vivo setting. So let's start off with our definition of a humanized mouse. We consider these animals basically immunodeficient mice that have been engrafted with human cells and tissues in such a way that first these human cells and tissues first survive the engraftment procedure, but more importantly, that they maintain overall functionality. And we view this as a preclinical bridge between work done on the benchtop and the laboratory, between work done in the clinic. Now, there are a number of applications of humanized mouse models in biomedical research, and highlighted on the slide are just some representative examples of these. For example, these animals are being used in regenerative medicine, where they can be used to test cell populations derived from induced pluripotent stem cells or embryonic stem cells for safety and efficacy for function in the mouse models. They can also be used to study hematological disorders in order to understand some of the basic components that are necessary for the development of immune cell populations, and also to examine the roles of specific genes and how they function in those immune cell populations. They can be used to study basic questions of human immunology, including transplant rejection, vaccine development. They are also being used to study beta cell or insulin-secreting beta cell biology. There are a number of studies using these animal models for infectious disease, using pathogens as varied as HIV, EBV, Salmonella typhi, and dengue virus. There are also a number of laboratories trying to create autoimmunity models in these humanized mice, and these have proven to be very challenging because this requires the most intimate contact between the developing immune system and the host. And in these cases, we have a mouse, stromal tissue, interacting with a human immune system. And the final area where these animals are being applied is in the cancer biology and be used to follow the growth of cancer cells in the animals, to identify cancer stem cells, and an emerging field is to be able to understand how a human immune system interacts with these human cancer cells. So the next question is, why do we need humanized mouse models? Well, as most people in the audience probably are aware, most experimental studies have been done in small animal rodent models. And while these have really laid the foundation for our basic understanding of biological processes, the results generated in these small animal rodent models do not always translate into humans. Moreover, the outcomes predicted by these murine studies are not always representative of the actual outcomes in humans. So overall, this will permit the study of human-specific infections and therapies. This has been really highlighted, the importance for new models to study human immunity, in a recent publication published in 2013 in PNAS, in which the genomic responses in mice were compared to human for inflammatory stimuli. And there were a number of differences were identified in terms of the type of genes that were regulated in mouse and human, the breadth of the genes that were regulated in mouse and human, and the duration of the inflammatory response between these two different models. And a number of differences are really highlighted in this and really emphasize the need for new model systems to be able to study human immunology. So our overall goal is to enable clinically relevant in vivo studies of human cells, tissues, and immune systems without putting the patient at risk, and really asking basic questions about how human biology functions in a living test tube in these humanized mouse models. So the next question is, how do we actually get human cells and tissues to engraft, survive, and maintain functionality in humanized mouse models? And what we really need to do is target both the mouse innate and adaptive immune system. So just to kind of provide an overview for what the general immune response is to an antigenic challenge, I've shown you a diagram here highlighting the aspects of an immune response to antigenic challenge. This really starts off with the innate immune response, and this is characterized by the production generally of inflammatory cytokines by cells such as dendritic cells, macrophage, mast cells, and other types of innate immune cells, which help to drive the overall response. It's also characterized by, in many cases, by the activation of natural killer cells, or NK cells. And this is important because NK cells are exquisitely good at killing and rejecting non-self tissues and prevent a significant barrier for humanization. Now, depending on the quality of the innate immune response, this then feeds into generation of the adaptive immune response. And this includes the generation of antigen-specific T-cell responses, which respond to the challenge, in many cases control that antigen load, or a viral infection or a bacterial infection, and then go on to form a long-term memory, which is the basis of vaccines. This also involves the generation of B-cell responses, which are able to produce antigen-specific antibodies and undergo class switching, producing antibodies such as IgM, G, and A. So for the ideal mouse model, we really need to target, again, both the innate and the adaptive immune system, either eliminate it or significantly reduce it. And this really all started with laboratories targeting the adaptive immune response, so taking out those T cells and B cells in the mice. And there are two ways in which we can target the adaptive immune response in mice or mammals, and these are listed below. The first is the severe combined immunodeficiency mutation. This is basically a mutation in the catalytic domain of a DNA-activated protein kinase that is required for non-homologous end-joining immunoglobulin genes and T-cell receptor gene rearrangements. So with this mutation, you no longer get VDJ recombination, no B-cell receptors or T-cell receptors, and no T-cells or B-cells. An important caveat for this is that SCID mutations are actually important for DNA repair, so SCID mice are radiosensitive. It's important to keep in mind whenever you're creating humanized mice, whether you have the SCID or number two, the RAG gene. So RAG1 and RAG2 are required for creating double-stranded breaks that are required for immunoglobulin and TCR gene rearrangements. So once again, no B-cell receptors, no T-cell receptors, no T's or B's. And differently from the SCID mutation, RAG mutations are not involved in DNA repair, and the RAG mutations are not radiosensitive. So the first initial studies, the groundwork for humanized mice, was the introduction of the CB17 SCID mouse in 1983. So the CB17 SCID mouse with the SCID mutation do not have mature T cells or B cells. Over time, you can see a little leakiness on the SCID background, but these are very low levels that can emerge as the mice age. And for all intents and purposes, these mice lack adaptive immunity. Now, the CB17 SCID can be engrafted with human PBMCs, HSCs, and human fetal tissue. But overall, it's generally very low levels of engraftment are found in the periphery of these animals. It's only usually between 0.01 to 0.1% of human cells detectable on a consistent basis in the periphery. Now, this low level of engraftment was really attributed to the high level of remaining innate immunity in the CB17 SCID mice, including natural killer cells, which are, again, really good at killing non-self tissues. So the next step on the road to humanized SCID mice was to take the SCID mutation and to cross it onto non-obese diabetic mouse background, or the NOD mouse. So as many of you know, the NOD mouse is a spontaneous model for type 1 diabetes. But with the SCID mutation, no T cells, no B cells, these mice no longer develop the autoimmune diabetes. So the NOD background also offers a number of genetic advantages which improves the engraftment of human cells and tissues and immune systems. For example, they have severe defects in innate immunity. They have reduced NK cell number and function, again, which I think is critical. They have impaired macrophage activation. they have defective DC maturation, and they also lack hemolytic complement. The NOD strain also has a polymorphism in the signal regulatory protein alpha gene that creates a more human-like bone marrow microenvironment for interaction with human hematopoietic stem cells, specifically through the interactions with CD47. The interaction between SERP-alpha expressed by phagocytic cells in the mice and human CD47 and the NOD background sends a good don't-eat-me signal to those phagocytic cells and allows a high level of engraftment of human cells and tissues. But even with all of this, the NOD-scid, we can increase the engraftment of human immune system and cells, but it still can be quite low, extremely variable, and it's still tough to get full functionality in these mouse models. Now, this low level of engraftment was still attributed to that residual innate immunity in these mice. And one other caveat for the NOD-scid model is they develop an IL-2-dependent thymic lymphoma as the mice age. So really you have anywhere from 9 to 11 months to actually use a NOD-scid mouse for an experiment. So the next step on the roadmap to create humanized mice was to take the NOD-scid animal and target the IL-2 receptor gamma gamma chain. So why the gamma gamma chain? So the gamma gamma chain is actually a component of high affinity cytokine binding and signaling for a number of cytokines which are critical in hematopoiesis, and these are listed below, including IL-2, 4, 7, 9, 15, and 21. And when you knock out the gamma gamma chain, you eliminate high affinity cytokine signaling for all of these cytokines. This by itself leads to defects in both innate and adaptive immunity. This is also the most common form of X-linked SCID disease, and shown in the picture to the left is David Vedder, the infamous bubble boy. So by targeting the gamma gamma chain, we've now created the animal known as the NOD-scid IL-2 receptor gamma gamma chain, or for short, the NSG mouse. Now, the NSG mouse has a complete absence of the gamma gamma chain. They now have a long lifespan because there is a defect in IL-2 signaling. These mice no longer develop IL-2-dependent thymic lymphomas. There's also further impairments in innate immunity compared to the NOD-scid, and probably the most important is the complete absence of natural killer cells, probably due to the lack of IL-15 signaling, which is critical for NK cell development. And we also have a variation in the NSG mouse, which instead of the SCID mutation, we have the RAG mutation. This is the NRG animal. So the NSG and NRG mice can engraft at high levels with human cells, tissues, and immune system, and it supports the engraftment of a wide variety of human cells and tissues, including tumors, human skin, human islets with hormone-secreting populations, human hepatocytes with specific mouse models, ES cells and iPS cells for regenerative medicine, hematopoietic stem cells, and mature human immune cells. Now, the NSG animal is one example of a gamma gamma chain deficient animal people use for humanization, but there are other alternatives that are available in the academic world. So we've already talked about the NSG mouse that was developed in Jackson Laboratories. There's also the NOG mouse, which is very similar. It's a NOD-scid background with a truncated version of the gamma gamma chain. So the NSG has a complete deletion, and the NOG mouse is a truncated version of the gamma gamma chain. There are also available a bulb RAG2 knockout gamma gamma chain deficiency, which we refer to as the BRG, which is often used at Yale and Zurich. And there's kind of an outbred H2D background mouse with the RAG and gamma gamma chain, which is often used at the Pasteur Institute in France. All of these animal strains will engraft with human cells and tissues, and you can generate human immune systems in these mice. But with our experience, we've had the best success in mice on the NOD background with either the SCID or the RAG mutation. So what I'm going to move to next is basically really now start to drill down into creating human immune system models with our humanized mice. And we've pretty much boiled this down to 3 basic models where we can study human immunobiology in these immunodeficient mice. And these are listed here. So the first is what we call the Hu-PBL-SCID mice. These are basically immunodeficient animals injected with human PBMCs. Now, for each one of these models, there are strengths and weaknesses, and I'll be describing some of these as I go through each one of these. So the advantage for the Hu-PBL-SCID mouse model is that this is probably the easiest to generate. Basically, you need to purchase or obtain human PBMCs, purify these cells over a glycol histopate gradient, inject those into the mice, and you'll get high-level human immune cell engraftment very easily in these mouse models. primary cell population, which engrafts in the Hu-PBL-SCID model, are T cells. You get very low levels of B cells engrafting and very few innate immune cells, but T cells are the dominant population that survive in these mice. One of the disadvantages or potential disadvantages for this mouse model is that T cells that engraft and survive are mature T cells, and they actually see the mouse antigens as xenogenic. So ultimately, these T cells will mediate a graft versus host disease and lead to the death of the mice. One of the other advantages for this model, as you can envision, is actually fairly straightforward to get clinically relevant samples, for example, from type 1 diabetics, MS patients, or rheumatoid arthritis patients, put those into the mice to study the biology of those T cells. The next model is what we refer to as the Hu-SRC-SCID mouse. So SRC are SCID repopulating cells, which are basically CD34 positive hematopoietic stem cells. So in this model, these are our immunodeficient mice that have been, in most cases, sublethally irradiated and injected with hematopoietic stem cells. These hematopoietic stem cells can come from a variety of sources, including cord blood, mobilized peripheral blood from adults, bone marrow samples, or human fetal liver specimens. The advantage of this model system is that when you engraft hematopoietic stem cells into our immunodeficient mice, you now grow a complete human immune system with T cells, B cells, and innate immune cells. One of the disadvantages for this model system includes the variety of ways that people actually assemble these mice in terms of injection routes, age of recipients, to generate a robust human immune system. And in these cases, T cell development in most cases is occurring on mouse thymic epithelium. So the majority of T cells which develop in this model are going to be restricted to mouse MHC. So this can create some problems when you try to generate robust host immunity in these mice, because the T cells will have difficulty talking to the human innate immune cells and to the B cells. So the final model really started out as the SCID Hu mouse, developed by Irv Weissman, Mike McCune, and Lenny Shultz. So in this model, these are basically our immunodeficient mice that have been engrafted with human fetal liver and thymus under the renal capsule. And when you do this, you actually grow a functional human thymus under that kidney capsule that can educate T cells. The problem with the base SCID Hu model is that it's very difficult to get robust peripheral engraftment of human immune cells with only that thymic organoid growing there. So this led now to development of what is most commonly called the BLT mouse for bone marrow, liver, and thymus. And for this, you do your thymic implant under the kidney capsule, but you also inject autologous CD34 hematopoietic stem cells from that same fetal liver. When you do this, the BLT mouse model is probably one of the most robust engrafting models that we have available in our humanized mouse repertoire. T cell development is very efficient in these animals and is occurring on human thymic epithelium. So the T cells should all be HLA restricted. A number of laboratories have used the BLT mouse model to actually study both immune responses and viral infections, for example, such as HIV. One of the disadvantages for the BLT system, and this can vary between laboratories, but in many cases, what we observe is a breakdown of peripheral tolerance as a BLT mouse ages. So usually within a number of weeks after implant and screening these animals, they also develop what looks like a wasting syndrome, which may be a graft versus host disease, but that's not exactly clear yet. But you also have a short window of time where you actually can study human immunity in the BLT mouse model. So what I'm going to do next is basically walk you through examples of each one of these three models and tell you a little bit about how we're using them to study human immunology. One of the issues with all of these models is it's still very hard to completely reproduce a human disease syndrome in these mouse models. But where they're most powerful is when you have a specific question or a specific immune cell population that you want to interrogate. There you can design focused experiments to address those questions and actually get some pretty powerful results. And I'll give you some examples of that as we move forward. So the first model to discuss is the Hu-PBL-SCID model. So again, a very basic model to set up where we start with our NSG or NRG animals. These animals can be preconditioned with the radiation or not preconditioned. We can then inject human PBMCs. This can also be human splenocytes into the mice. And we standardly, for our research, do 10 million, but you can do more, and you can also do less and get fairly good engraftment. We then can follow these animals for engraftment and disease. And we starts out as a nice, healthy mouse But over time, as these mice develop a xenogenetic graft versus host disease, they become sick, as you can see on the right. And this is characterized by hair loss and erythema, hunch posture, weight loss, and ultimately death. To give you a feeling for the kinetics of human immune cells in the PBL-SCID model, here we're looking in the peripheral blood of NSG mice that were injected IP with 10 million human PBMCs, looking at 2, 4, and 6 weeks after engraftment. And here we're staining the cells in the blood with human CD45, which stains most cells of hematopoietic origin. But again, in this model, the majority of these cells are going to be T cells. And you see as early as 2 weeks post-injection, we have a significant level of engraftment in these animals, and this continues to engraft over time. But as I stated earlier, ultimately these animals are developing this graft versus host disease and will succumb to that. The kinetics for the GVH onset are shown in the next slide. So here we're looking at cumulative survival over time, and with the solid line are NSG mice which have received irradiation only. And those animals do very well with a low dose of irradiation. When we only inject in PBL, which is the solid dashed line, you can see that over time, usually 4 to 6 weeks, these animals start to develop the graft versus host disease response and succumb to that GVH. If we combine irradiation and PBL, which is the smaller dashed line, we can see a rapid onset of GVH in these animals, and the majority of the animals also come to that GVH around the same time. So depending on the specific question that you have, you would either want to include irradiation if you want a faster onset of GVHD to potentially test therapeutics for slowing down the T-cell response, or if you're interested in evaluating more T-cell function, then without radiation will probably be the preferred way to go. But ultimately what we would like to do is be able to study T-cell function in the absence of the xenogenetic graft versus host disease in the PBL-SCID model. And we're developing models in collaboration with Jackson Laboratory to make this possible. So again, ideally we're studying human T-cell function in the absence of xeno-GVH. So to really make this happen, as I told you earlier, the majority of engrafted cells recovered from blood and spleen of NSG animals injected with human PBMCs are T cells. So our hypothesis was, in mice lacking murine MHC, they will have a reduced susceptibility to the xeno-GVH, because that's what we predicted to be the primary targets of the T cells. So we've generated a number of strains which lack MHC class 2, for example, the NSG ABO mouse, and mice which lack the expression of mouse class 1, either through the targeting the beta-2M allele or by directly knocking out the heavy chain in the KBDB null animal. And we know that really targeting that class 1 is important for slowing down that graft versus host disease, as shown in the next slide. So again, our basic setup for our Hu-PBL-SCID mouse model, we're looking at survival over time. In this case, the standard NSG mouse, which is the longer dashed line, succumbs to the infection pretty rapid, or the GVH fairly rapidly. Within 2 to 3 weeks, all the animals become sick. If we knock out class 2, which is the solid line and really relies on more of a CD8 effect, there's a slight delay in GVH, but overall, most of the animals are becoming sick very quickly in the class 2 knockout. However, if we target class 1, and this is shown in the smaller dashed line, we now significantly extend out the survival of those animals and increase the time where we can study T-cell functionality in those mice by 2 to 4 weeks, depending on the specific experiment. So again, by creating new mouse models that lack mouse MHC, specifically class 1, we can extend the utility of the Hu-PBL-SCID mouse model. So just to summarize that portion for the Hu-PBL-SCID description, we've documented that human PBMCs will induce a xenogenetic graft versus host disease in NSG animals. And we actually use that as a model to test T-cell specific therapeutics. I've also shown you that murine MHC is a primary target of the xenograft versus host disease, and that models lacking murine MHC can be used to study the effector function of human T cells in the absence of the xenogenetic response. Now, where we really like to move all these models would be to study human autoimmunity in these, and where we get PBMCs, for example, from type 1 diabetics and MS patients, and study their biology in these mouse models. So this brings on now to the second model that we use to study human immunology in these mice, which is the Hu-SRC-SCID mouse. Again, these are animals injected with hematopoietic stem cells and grow a complete human immune system. So what exactly is a hematopoietic stem cell? I'm sure everyone is fairly familiar with this, but basically, hematopoietic stem cells are a population of cells that primarily reside in bone marrow. They have a self-renewal capacity and maintain themselves at a very low frequency, but have the unique ability to differentiate into all cells of the hematopoietic system, including innate immune cells such as mast cells, dendritic cells, macrophage, natural killer cells. They also give rise to cell populations such as red blood cells, megakaryocytes, which produce platelets, and they give rise to the adaptive immune system, including T cells and B cells. And one of the important aspects for the development of this hematopoietic system is the reliance on specific cytokines to drive the differentiation of these specific cell populations. So again, one of the difficulties with the Hu-SRC-SCID model is how do you actually create and generate these mouse models? And there are a number of ways in which different labs actually do this, and a number of considerations that you need to think about when you go to set out to do this. So the first one is the age of the recipient. Do you use newborns, young adults, adult mice? Also the strain background. We talked about this earlier, where there are a number of common gamma chain deficiencies on different backgrounds that can be used to create humanized mice. The third is the source of human tissues and the cell dose. These hematopoietic stem cells can be derived from a variety of tissue types, as I mentioned earlier, cord blood, fetal liver, bone marrow, mobilized peripheral blood. And then you also have to think about your injection route. So the next slide basically outlines the two ways in which we engraft hematopoietic stem cells into our NSG or NRG animals. And we use both young adults and newborns. And our primary way that we like to engraft these hematopoietic stem cells is intravenously, tail vein into the adults, or by intracardiac injection into the newborns. Now, if you look in terms of the total immune system development in these mouse models, the newborns, both models engraft at very high levels with human immune systems following hematopoietic stem cell injection. But the newborns actually have a fast rate of T cell development, and this is slightly delayed in the young adult model. But basically the way we create these model systems is first giving a low dose whole body radiation. And again, it's important whether you're using the SCID or the RAG mutation that will dictate the dose of radiation that you use. For our laboratory, we use hematopoietic stem cells derived from CD3 depleted umbilical cord blood. We generally give anywhere from 50 to 100,000 CD34 cells injected into the mice, and we screen the peripheral blood generally between 12 to 16 weeks. But to give you a feel in the young 3 to 4 week old NSG animals in terms of engraftment and immune system development, the next slide basically gives you the engraftment levels over time in the blood of NSG animals, looking at total CD45. And you can see as early as 3 weeks post-injection of these animals, you can find human immune cells developing in the blood of these mice. This tends to peak out by 12 weeks and then be maintained out to 18 weeks. But as I mentioned earlier, T cell development in the adults is a little bit slower, these young adult NSG animals. So in the next slide, we're actually looking at T cell development in the peripheral blood, looking at CD3 positive T cells as a proportion of CD45 over time. And you see, even though we had robust engraftment by 12 weeks, T cell development is much slower in actually repopulating the periphery of these animals. And these animals really aren't useful, at least for our experiments, where you need robust T cell development until week 15. So again, depending on the model system that you're using and the engraftment criteria that you need to understand how these animals engraft and when's a good time that they can be used for your experimental protocols. So the data to this point really show that we can engraft human immune systems, including T cells, into these animal systems. So we really want to test now overall functionality. And for this, we want to look at the innate immune response and the adaptive immune response. So let's talk about the innate immune response first. And the way we actually wanted to examine the innate immune response that can be generated in these humanized mice is by stimulation of the innate immunity by lipopolysaccharide or LPS. Now, this diagram is a very, very simple overview for what happens with LPS signaling. It binds to TLR4 and a number of other modulatory proteins. It initiates 2 independent cascades of intracellular cytokine pathways, and this results in the release of inflammatory cytokines such as TNF, IL-1, IL-6, and IL-8, and the release of type 1 interferon. So what we wanted to do was to challenge our humanized mice injected with human hematopoietic stem cells and look at the innate immune response generated by that human immune system. But there's a problem with this approach, and that is that the mouse innate immune system still present in the NSG background will respond to LPS and result in the production of mouse cytokines, which may change the type of a human immune response that you have. So to get around this, we actually took the NSG animal and targeted the mouse TLR4 receptor. In theory, this should eliminate the mouse response and allow us to specifically study the human response. So we created this mouse, and as a proof of concept, we took unengrafted, so no human immune system NSG or NSG TLR4 animals, and we challenged them with LPS and looked at the production of mouse inflammatory cytokines, shown on the right. As you can see, the NSG animal actually can respond to LPS challenge, but an NSG animal lacking TLR4 is very ineffective at responding to TLR4. So now we have a mouse model where we can study the TLR4-mediated response by human cells in the absence of the mouse response. So for the basic experiment, we actually used our newborn engraftment model, again, low-dose whole-body radiation, hematopoietic stem cells derived from CD3-depleted umbilical cord blood. In this experiment, we injected 50,000 CD34 cells by intracardiac route into our newborn NSGs, or TLR4s. And I'm only going to show you the TLR4 data in this experiment. But we looked at the levels of human inflammatory cytokines in the serum of mice at 6, 12, and 24 hours post-LPS injection. Now, for simplicity's sake, I'm only showing you the responses to IL-6 and IL-8 in either mice that were challenged with PBS or LPS. And in this case, we also included a 2-hour time point. But here we're looking at the response to LPS at 2, 6, 12, and 24 hours at either IL-6 levels on the left or IL-8 levels on the right. And as you can see, we actually have a rapid cytokine response, human cytokine response in these mice, demonstrating that the human immune system is functional, and it then does tend to decrease very quickly, and by 24 hours, most of the cytokine response is now gone. But now we have a humanized mouse model that lacks the expression of mouse TLR4, where we can specifically interrogate a human innate immune response to TLR4 agonists. So the innate immune system is functional. How do we study the adaptive immune system? And for that, we turn to a transplantation model, because you know T cells and B cells are going to be actively involved in the rejection of transplanted tissues. So again, the transplant of non-self or allogeneic tissues induces a robust host immune response to those tissues and ultimately results in the rejection of those tissues. Now, in the clinic, we can prevent that transplant rejection with the use of immunosuppressive drugs. However, these are globally immunosuppressive and not only will prevent the rejection of the tissues, the transplanted tissues, but will prevent the ability of the host to respond to infectious disease or to malignancies. So the goal of transplantation immunologists are to develop protocols to specifically suppress the immune response to the allograft, what we call transplantation tolerance. Now, my laboratory and many, many other laboratories have got this to work really well in immunocompetent mouse. For example, shown here on the right is a bulb C mouse receiving a completely MHC mismatched skin graft from a B6 animal. And we get these animals to maintain that graft long term. However, these same protocols have not translated into human transplantation scenarios for reasons we don't completely understand. So we've been developing models where we can study allograft rejection in our humanized mice. And we have two ways in which we really do that. One is the rejection of human skin grafts on the animals, which I'll be showing some data today. But we also have models which might be more clinically relevant, looking at the rejection of human islets, which are being used for beta cell replacement therapies in diabetics. I won't be showing any of the islet data for sake of time sakes today, but really I'll be showing you a little bit of data on the rejection of human skin in our Hu-SRC-SCID mouse models. So one of the first things we needed to demonstrate was that human skin could actually survive on our NSG animals. And that's what's shown on the next slide, where we took a completely unengrafted NSG animal, transplanted human skin on these mice, and looked at survival over time. And as you can see in these different pictures I'm showing, we have a nice topical graft surviving on these mice. You can see well-formed by H&E staining epidermal and dermal layers on these mice. By CD45 staining, you can see actually pasture leukocytes surviving on these grafts. And you can actually see human vasculature developing by the CD31 stain on the lower right. So overall, the NSG animals will actually support robust human skin survival and will allow us now to ask questions: if we have a human immune system, would they be able to reject that skin? So here's the actual experiment Now, where we take, again, our newborn engrafted model, injected with 50,000 hematopoietic stem cells, we screen these animals for engraftment, and those which have an appropriate immune system, we can then give a split-thickness human skin graft and see if these animals reject their graft. This is kind of a summary topically of the data, where we looked at either an unengrafted NSG animal, and once again, you can see well-formed skin on these animals, including epidermal and dermal layers by H&E, and even with specific markers for the epidermis, involucrin, see nice staining, and for the dermis, including vimentin, we have a nicely formed dermis. On our HSC engrafted animals, what you're really left with, and these have a functional human immune system, is a scab-like structure on the animals with very little human epidermis or dermal layers detectable in these mice. And really, in the next slide, we look at just a summary of this data where we're comparing graft grades in unengrafted NSGs or HSC engrafted NSG animals. And what we find is the majority of HSC engrafted animals have a higher score for inflammation and necrosis compared to the NSG animals, suggesting that all these animals are rejecting their grafts compared to an unengrafted NSG mouse. So now we have a valid model system where we can test immune therapeutics, see if we can prevent skin graft rejection or change the immune response to that skin. And I'm going to present a little bit of data about how we're actually changing the nature of the immune response stimulated by that allogeneic human skin graft. And we really focused on the helper cell populations generated during the response to that human skin. So helper cell populations can represent a wide variety of human immune cells. So really, CD4 helper cells start out as a naive population with very limited functionality. But following antigen exposure and exposure to different kinds of cytokine environments, you can now give rise to a wide variety of helper cell populations, including the classical Th1, Th2s, some of the unique Th7, Th9 populations, also populations that have been involved with inflammatory disease processes in human skin, including Th17s and Th22s, also human Tregs, and follicular helper cells. So we wanted to ask the question if we really were generating human helper cell populations during the rejection of human skin. And for this, really focused in on CD4 T cells producing IL-17 and IL-22. So here we took the spleens from animals that had either not been exposed to a human skin graft on the left, or from two animals that were engrafted with hematopoietic stem cells and had rejected their skin. We recovered spleens from these mice, and we stimulated those cells in vitro with either PMA-Annamycin on the top row or no stimulation on the bottom row. And then we did a standard intracellular cytokine assay looking at CD4 CL cells producing either IL-17 or IL-22. As you can see on the left plot on top, we can actually find cytokine-producing populations in a more naive human immune system mouse. But when mice have rejected skin, what we find is actually the generation of cells that can produce IL-22, and these actually accumulate to a significant level, and in some cases, mice which are producing IL-17. This can be a little bit variable. For example, here we have one mouse, number 1, which actually had a significant IL-17 response, and one that really didn't. But overall, we're seeing the generation of these Th22 and Th17 populations of immune cells in these mice, which is exciting because we know these populations are actually involved in inflammatory disease processes in human skin. So this brings up the concept that we can actually ask a specific question in these mouse models, and can we change the generation of these helper cell populations in the mice? So just to remind you, the generation of T17s and Th22s is reliant on IL-6, and for Th17s on IL-23. So we actually used blocking reagents to eliminate IL-6 and IL-23 during the rejection of human skin grafts, and ask the question of whether we could change the way the human skin graft was rejected, and do we change the nature of the helper cell populations that we're generating. So here's the basic experiment where we took our HSC engrafted animals, they received a human skin graft, and then they were treated every 3 days with a bispecific reagent that actually blocked IL-6 and IL-23. We then evaluated the mice for skin graft survival and evaluated the T-cell response in the spleen of these animals. Now, I'm not going to show any of the skin graft rejection data because basically this treatment did not prevent skin graft rejection. The mice still all rejected their grafts. But what we did see is a change in the nature of the T-cell response. And that's shown on the next slide. So again, we're looking at our cytokine CD4 T cell populations after PMA Annamycin from mice that had rejected their grafts and received either no treatment or where we blocked IL-6 and IL-23 with our bispecific reagent. In the no treatment mice, we're showing two examples, but basically we have nice IL-22 and IL-17 producing populations in the spleens of those animals. But in mice that were treated with a blocking reagent for IL-6 and IL-23, we now completely prevented the development of those cytokine producing populations. So again, this demonstrates the power and utility of these mouse models where we can study immune therapeutics and change the nature of a human immune response. So just to summarize the data for the USRC-SCID mouse models, we've shown you that the humanized mice will produce human inflammatory cytokines in response to TLR agonists, demonstrating the functionality of the innate immune system and grafts in these animals. And the TLR4 mouse model really reduces that mouse response to LPS and allows us to specifically focus in on the human response. I've shown you that humanized NSG mice can reject allogeneic tissues by human immune cells, and now we can test therapeutics in that model. And finally, I've shown you that effector human T cells are generated during the rejection of human skin allografts in our HSC grafted animals. And then we can use, again, specific therapeutics to alter that human immune response. So now let's move on to the final model, which again is the BLT mouse model, which allows us the most robust engraftment for human immune systems. So this is basically how we set these animals up. Where on the top, I'm showing you that we take fetal thymus and fetal liver. We prepare 1 millimeter fragments of those tissues, and those get injected under the kidney capsule of our lately irradiated NSG animals. We are also at the same time preparing CD34 positive hematopoietic stem cells from that fetal liver and injecting 100 to 200,000 cells intravenously into those recipient mice. As I said, when you do this, you actually grow a functional human thymic organoid in these mice, and that's shown on the bottom left. At 16 weeks post-engraftment, remember we started with 1 millimeter fragments. We now have a substantially growing thymic organoid under the kidney capsule of these animals that can support human T cell development, and that's evident in the FACS plot to the right of that, where we have both double negative for CD4 and CD8, double positive cells, and the classical single positive cells that's characteristic of thymus development, of T cell development within the thymus. We also get robust peripheral engraftment of human immune systems in the BLTs, and that's shown in the next series of FACS plots. If we look in the blood of our BLT animals, we can find the development of significant populations of total human CD45 positive cells. Of these CD45 positive cells in the middle plot, you find both CD3 positive T cells and CD20 positive B cells. If you look specifically at those CD3 positive T cells, you can find both human CD4 and human CD8 T cells. And in the bottom plot, you can actually find human innate immune systems. It's kind of blocked out here, but you can find CD14, CD33 positive human monocyte macrophage populations. And you can find human 11C positive and human CD123 positive myeloid and plasmacytoid cell populations, respectively. So overall, we develop a great human immune system in these animals, including both the thymus, T cells that are HLA restricted by that thymus, B cells, and innate immune cells. So one of the areas where we're using this humanized mouse model is actually to study Tregs in these animals. And these mice also develop CD4 positive human Tregs, including FOXP3 positive CD25 positive cells. We've characterized these Tregs a little more substantially, showing they're also Helios positive and negative for CD127. So these mice not only develop your conventional T cells, they also develop regulatory T cell populations. So what we really wanted to do is not ask this BLT mouse model with a robust human immune system, can they respond to an infectious agent, such as dengue virus? Now, dengue has become really an important pathogen, both here in the U.S. and abroad. It's a mosquito-borne flavivirus. There are a number of individuals at risk worldwide and a number of deaths yearly. The problem with dengue is it generates a shock syndrome or hemorrhagic fever, which can have a high mortality rate in humans and children. It's often accompanied with a secondary infection of dengue virus, but the actual mechanisms of the disease process are not quite clear. It can be through antibody-mediated enhancement of infection or this through generalized cytokine release. But we really need a good model to understand how dengue virus causes these problems. So what we really wanted to do was to take our BLT mouse models and ask whether we can generate efficient T cell responses and B cell responses to dengue virus. And we can. So on the next slide, we're actually evaluating the generation of T cell responses, cytokine-producing CD8 T cells following dengue virus infection. And if we recover the spleens from animals that were infected 7 days previously and stimulate those with known A2 epitopes that should stimulate those CD8 T cells, we can actually find interferon gamma-producing CD8s and TNF-producing CD8s in response to those peptide stimulations from our dengue virus-infected BLTs, confirming that dengue virus infection will generate human dengue virus-specific CD8 T cell responses in these mice. We can also find antibody responses. This highlights one of the problems with these models, where we can actually find a good IgM response to the dengue virus envelope protein in infected animals, but it's very difficult in these humanized mice currently to get robust IgG responses. So basically, to summarize the BLT mouse model, I've shown you that the BLT model allows for robust and consistent engraftment of human cells, including multiple lineages. Probably most importantly, are HLA-restricted T cell development. I've shown you that we can generate efficiently human Tregs that are being educated on human thymic epithelium in these mouse models, and we're currently using this as a platform to test efficacy studies to boost human Treg number and function. And I've shown that we can generate dengue virus-specific immune responses in these mice, including epitope-specific CD8 T cells and virus-specific IgM, but not IgG in these mice. So on the final discussion slide is to really talk about the limitations of these humanized mouse models. A number of laboratories, including ours, have made significant progress in making these useful, but there's still room for improvement. And these are listed on the next slide. Basically, there's a lack of HLA molecules being expressed in these mice that are important for T cell development and for homeostasis. So a number of labs, including ours, have developed NSG animals or other strains expressing human HLA. There's also the concept that there's a species specificity for cytokines and growth factors that are important for hematopoiesis, and that the mouse versions do not act as effectively on the human cells. So a number of laboratories are now creating base strains of mice which express human cytokines that should be able to improve both total engraftment, T cell function, B cell function, and innate immune cell function. Most of these models also lack lymph node development due probably to the lack of IL-7, which inhibits the ability of lymphoid tissue initiating cells to develop. So we're hoping that transgenics will improve that. And while these animals are all severely immunocompetent, there still is residual mouse innate immunity in these models, and that by continuing to target mouse genes, we can severely depress that mouse innate immune system. And one of the final areas that we're working on is to improve the homing ability of these human cells in the mouse. And this will involve the expression of human integrins and chemokines in these mouse models. So overall, we think we are making a pretty good argument that humanized mice can be used for personalized medicine and will continue to improve as we make modifications to these mice. This is just to acknowledge some of the people in our laboratory who contribute to this work. And my collaborators at UMass are listed on the left and in the center. I'd also like to acknowledge Lenny Shultz at the Jackson Laboratory, who we work very closely with to create these humanized mice, and also Roland Tisch, who we're doing a number of diabetes studies with and Treg studies. And below are the basic funding sources for helping to support this work. Thank you for your attention.
Thanks for that really informative presentation. If you have any questions for Michael, I encourage you to submit them now. Both Michael and Charlie will be available after Charlie's presentation to answer your questions. Charlie, we're ready for you to begin.
Great. Thank you, Tamlin, and thank you, Michael. First, I'd like to thank the team at Gen for hosting this forum on what I think is some very exciting and enlightening work, and Dr. Brehm for the explanation of some of his groundbreaking research. For my part today, I'm just going to provide a brief summary of the resources that the Jackson Laboratory has available to support innovative research in basic science and drug discovery across a spectrum of therapeutic areas, and specifically the provision of some of the JAK's humanized NSG models that Dr. Brehm has just so eloquently described. If you're unfamiliar with the Jackson Laboratory, our headquarters is located in Bar Harbor, Maine. Since our founding in 1929 as a cancer research institute, JAKS has been a leading authority on mammalian genetics and human disease modeling in mice, and is the leader in the production of high-quality research mice and the development of cutting-edge research mice like the humanized mice that were described by Dr. Michael Brehm in his talk. Building on this foundation that JAX has continued to integrate basic research with human clinical applications. We just recently opened in October of 2014 the JAX Center for Genomic Medicine in Farmington, Connecticut. This facility is designed to keep JAX, our research scientists, and the entire scientific community really at the forefront of human genetic and genomics modeling, where our cutting-edge genetic and pharmacology services help to interpret genomic complexity and empower the drug discovery scientific community with the data, the tools, and the expertise that are needed to improve patient health. About 10 years ago, we opened our facility in Sacramento, California. This is a large mouse production and mouse custom breeding facility. This is also where we execute the development and production of advanced disease models like the humanized NSG mice of today's presentation, and humanized as well as non-humanized cancer models, including patient-derived xenograft or PDX models, and conduct drug efficacy studies in oncology, immunology, and neurology. With scientific partnering and research empowerment really being core to our institutional mission here at JAX, the Jackson Laboratory is the world's largest repository for mouse models of human disease. We import and cryopreserve as many mouse strains as possible to ensure their availability for all time to the scientific community. Among the more than 7,000 research strains that we currently distribute at both our Bar Harbor and Sacramento facilities, over 1,400 of those strains are actually maintained as live colonies. And as a result of the strong scientific partnering that we've established with research institutions and individual investigators across the world, JAX mice or J strains are the most published research strains currently in existence. The Jackson Laboratory distributes the best characterized, most genetically stable, common inbred strains like the B6J, the BALB-J, and the DBA2J. It's the use of these common inbred strains that supports the collection and distribution of specialty mutant mouse models and human disease, as well as provide funding for freely available online resources that we provide to scientific community. As Michael described, there are a variety of humanized mouse models that are useful for the study of many different types of human clinical conditions, including infectious diseases and for immune oncology drug design. The Jackson Laboratory specifically distributes 3 distinct models of humanized mice: humanized CD34 NSG mice that have been engrafted with human CD34 hematopoietic stem cells, the humanized BLT or Hu-PBL-SCID NSG mice, and then the Hu-PBMC NSG mice, which have been engrafted with peripheral blood mononuclear cells. And then we also conduct a variety of in vivo pharmacology studies in these mice as well. As the name implies, all of these models utilize the NSG mouse or the NOD-scid gamma model described by Dr. Brehm and developed by the Jackson Laboratory professor Lenny Shultz as the platform for humanization. These mice have been shown to support the highest engraftment of humanized stem cells of any mouse currently available and recapitulate the most functional human immune system to date. NSG mice can be obtained from JAX for self-engraftment in your laboratory or facility, or post-humanization, so following humanization by our staff here at JAX. JAX has been distributing humanized mouse models for many years, and all of our models are validated to have achieved threshold engraftment levels prior to shipment or being placed on study to ensure the highest quality in experimental application. Although Michael has discussed in much greater detail the various types of humanized models and the appropriate choice of model for your specific application, this slide summarizes briefly for your reference the process that JAX employs for the creation of each of the model types that we distribute and the advantages and considerations for using each of these models. For example, the BLT humanized mice have achieved high-level adoption in HIV research and efficacy testing as they successfully support and model mucosal-based infection and immunity of certain viruses, including HIV. As Michael alluded to, an approach that is rapidly gaining acceptance in modeling immunomodulatory drug responses is the engraftment of cancer cell lines as well as human tumors into humanized CD34 NSG and humanized BLT NSG mice. The Jackson Laboratory, as I mentioned, can not only distribute model cohorts directly to your facility from our portfolio of humanized NSG mice, but can also execute client drug studies utilizing these mice on your behalf. Over the past several years, we've developed extensive expertise using and executing studies with these mice on behalf of our clients, and our technical support teams and study directors can be helpful in making the most appropriate model selection and study design for your application. So with that, I'll go ahead and turn it back over to Tamlin.
Thanks, Charlie, for that quick overview. We'll definitely have plenty of time to answer your questions. So once again, I'm going to plug that live Q&A, which will commence in just a minute. But first, I want to let you know that a post-webinar survey will deploy shortly in the presentation manager. You might need to disable your pop-up blockers to see it, but we hope that you do when you take a few minutes to respond. Your comments are extremely valuable to us. All right, we're ready to start the Q&A session. We have a lot of good questions already, and we'll answer as many as we can.
Our first question is for Mike. Mike, where do you get fetal liver and thymus?
So currently there is one primary company that most laboratories making the SCID Hu or BLT model use, and that's Advanced Bioscience Resources for purchasing the fetal liver and fetal thymus.
Okay, that was quick. Any models for human DC?
So again, most of the models that are engrafted with HSCs will develop human dendritic cells, both myeloid and plasmacytoid populations. Now, those can be improved upon by the expression of human cytokines, such as cytokine like GM-CSF, which will drive myeloid dendritic cell development. Those can be either administered. There's been one publication demonstrating that the delivery of a plasmid expressing GM-CSF will boost the levels of myeloid DCs. And you can also find increased levels of myeloid DCs being CD11C positive in the NSG animals, which are expressing stem cell factor, IL-3, and GM-CSF. And I believe that's commercially available from Jackson Laboratories, the SGM3 mice. And that's been published to show significantly higher levels of human dendritic cells.
Okay, Charlie, maybe that's a perfect segue into the next question for you. Does any company provide this model for preclinical service?
So obviously my slides alluded to that a little bit. JAX does provide the 3 specific humanized models that I explained in my presentation, the CD34 engrafted mice, the BLT mice, as well as the PBMC mice. We make those available as animal cohorts, so cohorts of humanized mice that can then be shipped to your facility for execution of studies in your laboratory or facility. Or we can also execute studies in our own facility in Sacramento on behalf of clients as well.
All right, Charlie, I have 2 other questions for you that are similar vein. What are the engraftment levels of human cells in Jackson humanized mice, and also what is the availability of your inventoried mice?
Sure. So the engraftment levels for all of the mice that the humanized mice that we sell meet a minimum threshold of 25% human cells. Often the engraftment is much higher than that, but any mice that we bring on study in our facility or ship out to our clients meet that 25% minimum threshold for our CD34 or BLT models. And then as far as availability and inventory, our CD34 engrafted mice are maintained as on-the-shelf inventory, so we maintain high levels of inventory for those mice. Often mice are available from the time of inquiry or order placement, but at a maximum they'll be available within 12 to 15 weeks post order. And then the BLT mice and the PBMC mice are produced more on demand.
All right, Mike, we're back to you with a couple questions. Do NSG mice engrafted with human HSC support the development of human RBC and platelets?
So with most currently available models, we'll find progenitors for platelets and RBCs in the bone marrow. However, it's very difficult for those cells to actually, the mature forms, to reach the periphery. And one of the primary reasons for that is that mouse macrophage are actually preventing the development of those mature cells in the periphery. So in a series of publications, depletion of mouse macrophages will increase the development of those peripheral RBCs and platelet levels in the mice.
All right, Mike, here's another one. Is human immune system development comparable between the NSG and NRG mice?
In all of our testing, the levels are fairly similar between the NSG and the NRG mice. The main difference is the radiosensitivity of the NOD-scid model, NSG model, versus the NRG model. So that's just a really important caveat for that. If you use the NRG mouse, you can use a higher level of irradiation.
All right, here's another one. For you, Mike, is the inability of human NSG BLT mice to generate antiviral IgG responses related to the maturation levels of the immune system?
The easy answer for that is we don't really know why most of the engraftment models, it's very difficult to get class switch definition matured antibody responses. We think it may be related to the maturity level of the B cells, which appear to be kind of stuck in either an immature or a transitional status. But there are also potential other reasons why the antibody response is kind of limited in most of these models. It could be the lack of lymph nodes, the lack of germinal center development, or the inappropriate communication between the T cells and the B cells.
Okay, thanks, Mike. Unfortunately, our time has run up. Thanks for listening in, and thanks for the great questions today. I'm sorry we could not answer all of them. This webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and colleagues. Once again, I want to thank our panelists as well as our sponsor, the Jackson Laboratory.